All right, so today, go ahead and then begin our lecture on blood vessels and hemodynamics. First couple slides, just like I did with the heart, going to be a little bit of anatomy-based stuff, but as you know, we talked about the heart, anatomy affects function, and we've talked about that since the beginning of the semester. We're not really going to get into individual arteries and veins. We talked really in this class about a couple of arteries and veins, which were associated with the heart. We don't get into any of these really that are labeled here in this figure. I didn't really have a good figure to demonstrate blood vessels, so I just do this one in from your textbook. We're going to spend the majority of our time today talking about various hemodynamic factors. So we're not going to be talking about where blood flows. We're going to be talking about how blood flows, amounts, and what happens when blood flows, which is what hemodynamics are. And so we'll be talking about things like capillary exchange and venous return, blood pressure, which everybody's aware of, I'm sure, and quite a few others as well. So we'll be talking about that. And then the very end of today, we'll probably just get to start to talk about how we control blood flow, which relates to stuff that we talked about the other day with our hearts, and that can all be traced back to our brain, specifically that medulla oblongata, which we've talked about for quite a while now. So, first couple slides here, like I said, are anatomy-based stuff. Those of you who have taken anatomy with me, it should be a nice review for you. For those of you who are in anatomy this semester, we'll get to these things next week. Well, our blood vessels, as we've talked about, are a component of our cardiovascular system, along with our heart and our blood, two other lectures we've already talked about now. Our heart and our blood vessels provide avenues for the flow of blood. And we don't just pump blood because it's fun to do. We pump blood for the exchange of nutrients and to remove wastes. And because, as we know, most of the cells of our body cannot move around to get the nutrients they need, and they cannot move to deliver the waste to our lungs to breathe off CO2 or to our kidneys to get rid of things like urea and uric acid. Blood vessels are the avenue and extensions basically of our heart for the flow of blood to those cells, either to distribute the nutrients they need or to remove the wastes that they produce. Blood vessels also play an important role in adjusting velocity, which we've talked about velocity today, the speed at which blood flows, and then volume, which refers to the amount of blood that we have. And so affects volume and velocity of blood flow. There are kind of three major, in my opinion, blood vessels: arteries, veins, and capillaries. There's a figure actually here on 2 slides from now that show those three. The book, for whatever reason, kind of breaks it down to 5. They throw in arterioles and venules. Arterioles are a smaller form of artery. They're the smallest, actually, and venules are the smallest form of veins. I get why they do it, but to me, I was really kind of emphasize arteries, veins, and capillaries. We've already talked a little bit about arteries and veins. Remember, arteries are the blood vessels that carry blood away from the heart. We typically describe that blood as being oxygenated, like your aorta that we talked about associated with the heart that receives blood from the left ventricle. There is an exception to that oxygenated rule, which were the pulmonary arteries and pulmonary trunk, which receive blood from the right ventricle, deliver blood to your lungs. Okay, we learned that those were the only blood vessels that carry they're the only arteries that carry deoxygenated blood. I don't really like the term deoxygenated blood because it implies, in my opinion, that the blood is not containing any oxygen. And that's not actually the case. And even when you are at maximal exercise and your body is really extracting oxygen from the blood that's coming back by way of your veins, like your vena cava that we talked about the other day, to your right side of the heart, still actually contains oxygen. Like right now at rest, the blood that's coming back to your right atrium from your systemic circulation still is probably about 75% oxygenated. So deoxygenated really is kind of a term that really is misleading in my opinion. So it's just less oxygen than what our arteries typically will have. Capillaries are where the magic of the cardiovascular system takes place. They're the site of exchange. We'll look at their structure. They're very thin wall, which allows them to enhance exchange. Okay? They're found between our arterioles, which are the smallest arteries. They form from larger arteries and medium-sized arteries. And they're also found on the other side by or between venules, okay? Small veins collecting blood from capillaries, delivering them to the larger veins, which are the blood vessels, as we learned, carry blood towards the heart, typically being deoxygenated, or as I like to say, a little bit less oxygenated. Looking at the structure of our blood vessels, arteries and veins are very similarly structured. They both have 3 tunics or layers. The word tunics is a fancy word to describe layer. Okay. There's the tunica intima or interna, the innermost layer. Okay. There's several layers actually of them. Okay. Sublayers, as we could call them. The tunica media, which is the medial or middle layer. Okay, usually is the thickest. This is where you find that smooth muscle, and we know smooth muscle, based on what we've learned before, is involuntarily controlled by way of that autonomic nervous system, and is going to route blood flow by way of constricting or dilating. Okay, when the muscles contract, they constrict, which means blood vessels will decrease their lumen size, therefore blood flow decreases through them, and then they can dilate or contract or relax their muscle, which increases the amount of blood that flows through them. The least important ones, I should say, the least important layer for us anyway in this class is the externa, which is basically just connective tissue that kind of holds blood vessels in place. And what's actually cool is your larger arteries and veins will actually have blood vessels within this layer. So there's blood vessels within blood vessels, which is kind of cool. Here on the next page, I want to talk about those 2 arteries and veins. Okay. First, I want to talk about the interna. Okay. It's made up of kind of 3 layers. The one that we're going to focus on is the endothelium. Endothelium is the innermost part of the blood vessels, and it's actually found in all 3 forms: arteries, veins, and capillaries. It's a very thin, it's only 1 cell layer thick, and the cells are flat and plate-like, okay, surrounded by a basement membrane. You can see that in all 3 images. Okay. You'll notice on veins, they have inner foldings of that endothelium that form valves. And those valves are like the valves that we talked about found in our heart. They're also like the valves that are found in our lymphatic vessels, which allow, in this case, blood to only go through in one direction, never, at least hopefully, backwards. Okay? And like I said, those are influenced by or formed by the endothelium. If you look at the smooth muscle and the tunica media, which I want to talk about now, you'll see a lot in the arteries, very, very little in the veins. That's very important because arteries are really the directional controlling blood vessels, okay, controlling where the blood flows. For example, like when we want to exercise, we route blood to our heart muscle and our skeletal muscle. We've talked about that before. And we cut it off from places like our stomach and our kidneys. So in that case, we would perform vasoconstriction, and in our blood vessels, we would be dilating the arteries that feed those. Okay. Now, since arteries have a very thick layer of smooth muscle, they're going to actually have a much smaller lumen than what we see with veins. Therefore, veins can actually store and hold a lot more blood than your arteries. And that's going to relate to our first hemodynamic factor, what we call capacitance. And that's going to be talked about here on the very next page. Now, veins can also constrict their blood vessels, but when we use the word vasoconstrict, we're really actually referring to arteries, because the word venoconstrict refers to veins. Now, down at the bottom is an image demonstrating a capillary. Like I said, capillaries are thin-walled. They only have that endothelium and the basement membrane, which is extracellular material surrounding it. And they are thin-walled in order to promote stuff either entering the blood or exiting the blood. And then for stuff to either enter or exit the blood, they actually have to travel through the walls. Now sometimes there's little holes and little gaps for them to go. In some cases they actually have to travel directly through the cells. Okay? So I would hope you guys can see that it would be a heck of a lot easier to travel through a couple layers, like we have down here, versus several layers here and here. And so we'll get to capillary exchange, another hemodynamic factor, in just a little bit. Okay, our first hemodynamic factor is this capacitance I was just talking about, or venous capacitance, kind of be more specific, which relates to that large lumen you'll find in your veins. Because it has a relatively small, or because they have a relatively small amount of smooth muscle, they can accommodate large amounts of blood without changing our blood pressure, which is great. This property, capacitance or venous capacitance, allows veins to act as a reservoir of blood, okay, or for blood. And we can see that here with our very simplistic demonstration or illustration of the cardiovascular system. Here's our heart, here's our arterial circulation, here's our capillaries. Okay, arteries, are going to have, not a great ability to balloon out, nor will capillaries, but veins do. They say at rest, about 64% of your blood is stored, I'm going to use air quotes to describe stored, in our systemic veins and venules. Okay, so over half, and it's quite a bit more than half. Now, when we need to redistribute blood, like in cases during exercise, or if we're running from a bear, a fight or flight situation, what do you think that number is going to do? Is 64% going to increase, or is it going to decrease? Are we going to increase the amount of blood that we keep in our veins, or are we going to decrease it? We need to get it to the things that are working, our skeletal muscle, so we're going to have to decrease it, right? So the pie piece here would go much smaller. Okay? And that, like I said, is accomplished by venoconstriction. Okay, not as dramatic as vasoconstriction, but it does constrict veins, reducing the volume of blood in the reservoirs, allowing a greater volume of blood to flow to the needed tissues, i.e. skeletal muscle during exercise. This is activated by the sympathetic nervous system, and that is controlled by way of that cardiovascular center in our medulla oblongata, the brainstem structure that we talked about the other day and have talked about for several weeks now. This venous capacitance and venoconstriction, as we'll talk about in a little bit, is going to increase venous return, which we mentioned venous return the other day, something that actually influenced preload, one of those factors that influenced stroke volume. So there's all sorts of different terms that you guys are learning about now, and that's cool, hopefully, for you guys. I mentioned a little bit ago that the whole reason that we have blood, our heart, and our blood vessels is to distribute the nutrients and remove waste from those cells that cannot move. And that takes place within our capillaries through a process we call capillary exchange, okay, which is the movement of substances between our blood and interstitial fluid, and that can be from the blood into the interstitial fluid or from the interstitial fluid into the blood, okay, because it can be either or direction. Okay. Three different means of which are utilized for this to occur. The first and most important method is just diffusion. Diffusion is a term that we've talked about since the beginning of the semester, where substances move down their concentration gradients from high to low. We talked about like simple diffusion, where things can go directly through a cell. We talked about facilitated diffusion, where they can go through the plasma membrane, but they need a protein to help them get there. And There's also some other ways that stuff can go, which is not directly through the cell. Okay. Diffusion involves the crossing of a capillary wall either through intercellular clefts, which are very, very tiny gaps between the cells that form the capillary. They can also go through fenestrations, which are little holes that are found in capillaries, or they can just directly diffuse through the endothelial cells. And here, a couple slides from now, I can show an image to just demonstrate what I'm talking about. Okay. Down here at the bottom, we're showing water, okay, how it can actually pass, in this case, directly through the cell that's shown down here at the bottom of our capillary. There looks like 3 cells forming our capillary here. Some things, you can see oxygen, carbon dioxide, steroid hormones easily cross directly through. That's simple diffusion going directly through the cell. Some things go through the little gaps between cells, what we call intercellular clefts. Inter, remember, means between. Intercellular means between cells. Clefts are just tiny little gaps. And we're talking itty bitty small gaps. Okay. And then fenestrations, you can see I've got a picture. I don't want to get really into it in this class, but some of our capillaries are described as fenestrated. That's what these are right here. Little big for me. I can't really see it greatly here because I'm not zoomed in, but some capillaries actually just have little holes in them. Okay. And here in a second I'm going to talk about these that are really holy, which are sinusoids, which we find in places like our bone marrow and our liver spleen. Those sinusoids, like I was just saying, have so large of holes, basically looks like Swiss cheese, that those red blood cells that are formed, white blood cells that are formed, platelets that are formed, even the plasma proteins that we make in our liver can directly enter or exit our bloodstream. And that's what you can see there on the bottom of that figure, or on the bottom of that page, which are the leakiest of our capillaries. But we don't find those in very many places. Really just our red bone marrow, our liver, and our spleen. The second form of capillary exchange is by way of transcytosis. This is the process that we actually talked about back when we first learned about cells, and we barely mentioned it. I mentioned that we really wouldn't talk about it in this class very much. This is really that very little time. The third is called bulk flow. This is where we move a whole bunch of things, and that's why I call it bulk flow. It's moving in bulk. Let you guys finish writing, and then I'll talk about what I'm talking about here. Okay, so looking at the right side of our figure here, where we show a protein hormone like insulin that was listed as in our example of transcytosis. Transcytosis is a process in which a substance is brought in to the cell by way of endocytosis. We've talked a lot lately about phagocytosis, which is where those like macrophages can swallow up a substance to help digest them inside. Okay, phagocytosis is a form of endocytosis. Endocytosis is where we pull something in to a cell, forming a little bubble or vesicle, which is done when the plasma membrane surrounds the substance and pinches off to form the bubble around the substance, in this case a protein hormone. So that's endocytosis, shown right here. And then exocytosis on the other side, where the substance is being spit out. And it's basically just the reverse of endocytosis. If you remember, the prefix trans is a prefix that means across or through. Okay? So transcytosis is where we're pushing something through the cell, both by endo and exocytosis. It's important for mainly large lipid insoluble molecules that cannot cross the capillary wall any other way, like insulin as it enters our bloodstream. Bulk flow is, like I said, where we move a large number of things together in the same direction, things like ions, molecules, or particles. And it relates to pressure gradients, which we've learned a lot about pressure gradients here lately, from areas of high pressure to low pressure. This is a very important means of regulating volumes of blood and our interstitial fluid. We're going to talk a lot about bulk flow when we get to our kidneys, which is where we filter our blood and therefore control how much blood we have, which also influences, as we know, interstitial fluid amounts. The total pressure involved in this is described as net filtration pressure, or NFP. This is a balance of several other pressures, so it's actually a calculation, determining whether our volumes of blood interstitial will remain steady or change. Here in our next unit, we're going to talk about the word absorption, which is where stuff first enters our bloodstream. For example, the water that's in my cup over there that I'm drinking this morning is going to travel into my body through the walls of my intestine into my blood. That's what absorption is. That water that will then make its way into my plasma will eventually make its way to my kidneys and then get filtered. And it will leave my bloodstream and my kidneys, but it might then come back into my bloodstream. And that process is called reabsorption. When you redo something, you're doing something a second time. This time now we're reabsorbing. We're absorbing for a second time. Okay. That's a pressure-driven movement from the interstitial fluid, like the spaces inside of our kidneys, back into the blood. And for this to occur, we need a negative NFP. And so we can show that here with our arrow and NFP inside, moving into the bloodstream. The opposite of reabsorption is secretion, which is where, or excuse me, not secretion, but filtration, where the stuff leaves the bloodstream. And what we actually call the fluid that's formed when we're making urine, before it becomes urine, we call it filtrate. So we'll get to this in greater detail here in a couple weeks. All right, so the other day, when we talked about the heart, there was a slide that said, how does blood move? And in parentheses, part 1. Here comes part 2 right here. We talked about pressure gradients relating to flow of blood out of the heart. With the heart, it was actually really simple. In order for blood to leave our ventricles must generate more pressure than the pressure outside of them. For example, your left ventricle must exceed the pressure in your aorta for the aortic valve to open. In our blood vessels, or in our circulation, as we'll say here, there's tubes or tubings that influence the flow of blood, and therefore additional factor is present. There's an opposition to the flow of fluid through a tube we call resistance. Okay. Resistance has the word resist The word resist means to oppose. So this is a term that refers to the opposition of flow. Okay. In our cardiovascular system, opposition of blood flow in a vessel due to friction between, or is due to friction between blood and the walls of blood vessels is called vascular resistance. And vascular resistance is dependent upon three things. The size of the blood vessel lumen. Okay, remember that's the space on the inside where the blood is actually at. The blood viscosity, we used the word viscosity the other day. You remember what that means? The thickness. And then the total blood vessel length, so how long your blood vessels are totally. We mentioned something like 75,000. Now, of those three, we can combine them to form an equation. Not that you're going to have to do any math or anything like that, but it's 8 times viscosity, or thickness, times tube length, over, and this is the symbol in my easy way to do it, is pi. So maybe make it a little fancier on top. That's pi, 3.14, yada, 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 yada. Times radius, but radius is taken to the 4th power. And radius is halfway across the lumen. So when we say radius to the 4th power, we're referring to the lumen size. Now, let's think about those three factors. And which of those three factors is most influential on a second-to-second, minute-to-minute? Do we change our viscosity or thickness of the blood in a quick means easily? No, it's not like we can make our blood thicker or thinner really, really quickly. How about blood vessels and their length? Do they change length very, very easily? Or is that a very progressive thing over time? But what about the size of the lumen? Can we do this really easily? And look what happens when you multiply it to the 4th power. You're not multiplying it 4 times. You're multiplying it by itself 4 times. So you're not multiplying by 4, you're multiplying by itself 4 times. So changes in the radius or the blood vessel lumen can be very influential on the resistance. And therefore, resistance, as we said, affects flow. And we can describe the relationship between blood flow and resistance as an inverse relationship. When you say inverse, that means if one goes up, the other one goes down. So if there's an increased resistance, there's a decreased blood flow. Decreased resistance, increased blood flow. So let me read off, and I already talked about a lot of this just a second ago. Since blood viscosity doesn't change in healthy individuals, nor does the length of their blood vessels, the only method the cardiovascular system has to regulate vascular resistance is through changes in vessel diameter. Notice that since vessel radius is to the 4th power, and that's what we showed here on the previous page, small change in radius translates to a very large change in resistance. The smaller the lumen of the blood vessel, the greater its resistance to blood flow. So like our arterioles, they actually describe arterioles as resistance vessels because they have the greatest resistance. Now, you might think that's a bad thing because it sounds like they resist flow, but they actually are what control and determine where blood goes, which is important. Okay? Blood viscosity is not going to change, but it will. It's not typically going to change, but it will in responses to things like dehydration and polycythemia. Remember, polycythemia is like when people blood dope. They have too many red blood cells, and their viscosity is going to go up. Anemia, which is where they might have too many or too few red blood cells. Hemorrhage, they just have too little blood. They've bled. That's what hemorrhage is. That decreases viscosity. And then obese people, so people that are heavily overweight, often have hypertension. What's hypertension? High blood pressure. Okay. This is because fat is living tissue, in living tissue needs blood. And how does it get blood? By way of blood vessels. So people that have more fat have more blood vessels, therefore increasing their blood vessel length. And as I mentioned, blood flow is inversely related to vascular resistance. Way that can be, or two different ways to calculate this is by taking pressure 1 minus pressure 2 divided by vascular resistance, or by combining pressure 1 and pressure 2 to get mean arterial pressure. And I'll talk about that here in a second, divided by vascular resistance. Our total vascular resistance by all of our systemic blood vessels is defined as systemic vascular resistance, or just TPR, total peripheral resistance. As I mentioned, the smaller vessels, especially arterioles, but also your venules and capillaries, offer the most SVR. Our arterioles, like I said, are called resistance vessels. They have a major function to control SVR, which also influences, as we'll see, blood pressure and blood flow. We just talked about blood flow, by vasodilation and vasoconstriction. Major center for regulating it is the vasomotor center in our brain stem, our medulla. And that's the other part of the cardiovascular system. Last time we talked more about the cardiac center, regulating heart, things like stroke volume, heart rate. Here we're talking about vasomotor, controlling dilation and constriction. If vascular resistance is high, the only way to maintain blood flow is by increasing the pressure gradient. And this is something I always like to really emphasize. If you have high resistance, like high blood pressure, the only way to combat that is by making the heart generate more pressure with each contraction. So what's that mean the heart has to do? Pump harder. And that makes it very difficult over time on the heart. It makes it very hard. Who wants to work harder than they have to? Who wants their heart to work harder than it has to? Guess what happens to the heart if it gets overworked? It starts becoming ineffective. Down here at the bottom, I can demonstrate this concept of vascular resistance with three different blood vessels, A, B, and C. Okay, we're just going to use, and we're not going to use any units. We're just going to use 1, 1/2, 1/4. Okay, the radius of these three blood vessels. You guys would probably all agree with me that blood vessel A is 4 times larger than blood vessel C, right? So if you think about it, you would probably assume, had you not learned what you just learned, that blood vessel C would have 4 times as much resistance. That's not the case. Okay, because since we take the radius to the 4th power, look at the size differential, or the difference between the resistances. This has 256 times more resistance than blood vessel A. That's not 4 times, is it? It's a hell of a lot more than 4 times. That, therefore, then also influences the amount of flow. And this one can't hold as much as this one. Can't hold nearly as much as this one. Make sense? Okay, so we've been talking about blood pressure. Blood pressure is due to the contraction of our ventricles. As our heart pumps, it creates pressure in our blood vessels. Okay. That's what blood pressure is. It's hydrostatic pressure exerted by blood on the walls of a blood vessel. It's determined by cardiac output. What's cardiac output? Remember, it's the amount of blood your heart pumps per minute, influenced by how often your heart beats, what we call heart rate, and how much it pumps each time, what we call stroke volume. It's also affected by total blood volume. How much? The more blood we have, the more pressure. Okay? Think about it like a balloon. If you fill it up with more water, it's going to be more pressure inside, more likely to pop. And then vascular resistance, what we just talked about. Over here on the right, we demonstrate what happens to pressure the farther we get from the left ventricle. Okay, the very first blood vessel that receives blood from our left ventricle is our aorta. Look what the pressure does. It drastically decreases the farther away we get. Now, don't think measurable distance, think distance traveled. Okay, because your vena cavas are pretty darn close. They're actually right next to your aorta, but they're the farthest away in distance traveled, because your vena cavas deliver blood back to your right atrium. The whole reason is to help promote venous return. And so we'll see how this influences venous return here in a second. As most of you guys are aware, generally there are two numbers given when somebody gets a blood pressure taken. For example, 120/80. 120 would be the systolic blood pressure. Represents the pressure in your arteries and veins. We're typically measuring your arteries here. Brachial artery typically is the most often taken when your heart is in contraction, what we call systole, as we learned the other day. The lower number is the diastolic number when your heart is in diastole, or relaxation. You can combine those two and come up with mean arterial pressure. Now in math, what are you calculating when you calculate mean? And that gives you their average, right? So let's use 120/80. Okay, 120 plus 80 is 200. There's 2, so you divide it by 2. Gives you an average of 100, right? You would think that the mean arterial pressure of 120/80 would be 100, but it's not. It's about a third between DBP and SBP, because we actually spend 2/3 of our heart cycle relaxed. So your blood vessels are typically more often in diastole than your systole. So that's where this 1/3 comes in. So in that case, it's more like 92, 93, if I'm thinking right off the top of my head. So that's what mean arterial pressure is. You never have to calculate it or do anything like that, but in case you ever come across that. The great thing is, a lot of machines, since a lot of blood pressure cuffs are automated anymore, it's going to read that off just like it will your SBP and DBP. Okay. So, mean arterial pressure influenced. Two major things. Cardiac output. No cardiac output is influenced by heart rate, stroke volume. We learned the other day several different things influence heart rates. Epinephrine, norepinephrine, sympathetic activity. Decreased parasympathetic. Stroke volume influenced by afterload, contractility, preload. Okay, we learned preload was affected by venous return. We're going to talk more about venous return and the factors relating to it. I think that's the very next slide. Vascular resistance influenced by the three things we just talked about: total blood vessel length, viscosity of the blood, and then the blood vessel radius, or the lumen size. Okay, venous return, something that we said the other day affects preload. Venous return is exactly what it sounds like. It's the amount of or volume of blood returning back to our heart by way of our systemic veins. We're talking here returning to our right atrium and right ventricle. This occurs due to several different things. Okay. The pressure generated by constriction of the left ventricle is way up here, and then we have that progressive drop from there. Okay. Even in your venules to your vena cavas, and then into your right atrium and right ventricle. It's not a huge drop. Okay. If we go back to this figure, there's not a big difference throughout. Okay. Here's our smallest veins. Here's our vena cavas. We're talking 16, the max, all the way down to 0. It's not a huge difference, but it is enough of a pressure gradient to establish venous return. Okay. Besides that, there are besides the heart, there's two other pumps help do this, venous return. We talked about these the other day. The lymphatic system utilizes these the same way that your cardiovascular system utilizes them. You have the skeletal muscle pump, when your muscles pump, they eject blood, okay? And you can see in the figure that there are those valves. They prevent blood from going backwards, just like the valves we talked about the other day in your heart. And then the respiratory pump utilizes the changes in pressure that occur in your thoracic and abdominal cavities as we breathe in and out. Specifically, when we breathe in, it's because pressure in your thoracic cavity drops, abdominal cavity pressure goes up. That helps draw blood up, just like it helped draw lymph up. And there's a fifth factor, and that's the venoconstriction that we talked about earlier, which helps decrease venous volume, helping to return blood back to your heart. And those 5 factors, as we can see over here, influence venous return. Venous pressure gradient, venous valves, skeletal muscle pump, respiratory pump, venoconstriction that we talked about earlier. I believe this is our very last hemodynamic factor. It's one I have a fun analogy for. And this is another example of an inverse relationship, where once again, if something goes up, the other one has to go down. Or if something goes down, the other one goes up. When we talk about velocity of blood flow, we're talking about how fast the blood is flowing. Okay. And we're going to look mainly at our 3 major blood vessels, our arteries, our capillaries, and our veins. Okay. Our arteries and our veins, as you can see here, have a pretty high velocity, especially your arteries, which would make sense because they're the ones directly receiving the blood from your heart. Okay. Look at the capillaries. Venules a little bit, but mainly the capillaries is what I want to focus on. They have a very slow flow, but they also have a very large cross-sectional area. Cross-sectional area in this case refers to the amount of blood vessels. We mentioned the other day that there was a heck of a lot of capillaries in our body. They make up the majority of our blood vessels. Okay. So they have a very high cross-sectional area. Therefore, velocity is low. Now, the analogy I like to use is the same one that my professor at K-State that taught me this for the first time used. And that's the analogy of what a lot of K-State students like to do. Go to Aggieville, the bar district in Manhattan, do what college kids like to do, and then after, get picked up and go to McDonald's and get some food. Okay, that's better than a Big Mac after a bunch of beer. Okay. So, he always said, get picked up, driving to McDonald's is like blood flowing through your arteries. Okay. Go on the speed limit, hopefully, not being too reckless. If you get to McDonald's, are you going to travel the same speed through the drive-thru as you did to get there? No. McDonald's is like, especially the drive-thru anyway, it's like the capillaries. You have to slow down, you have to place your order, you have to pay, and in exchange for paying, you get your food. Because in our capillaries, we do the same thing, except for we're not exchanging money and food, we're exchanging nutrients and waste. And then when you leave McDonald's drive-thru, you hop back on the highway, just like your blood's going to hop back into your veins, and you're going to drive fast so you can get home and you can eat it. It's an easy analogy, worked really well for me. Hopefully it works well for you guys. Okay, the last thing I want to talk about, and this is what we'll actually spend pretty much the rest of the lecture talking about, which is how we control the flow of blood. Just like we talked about the control of your heart, it can be traced back to your cardiovascular center in your medulla. We learned on Wednesday how your cardiovascular center regulates heart rate and stroke volume. Therefore, since heart rate and stroke volume are the 2 factors that influence cardiac output, because remember HR times SV equals cardiac output also influences blood pressure. So we'll be talking a little bit more about heart rate and stroke volume in this lecture as well. Okay. Cardiovascular center also controls neural, hormonal, and local negative feedback systems, that can regulate blood pressure and blood flow to specific tissues. We have groups of neurons scattered within the cardiovascular center that regulate heart rate, contractility of our ventricles, and blood vessel diameter. So we're going to be talking about cardiostimulatory neurons, those that stimulate the neuron, or stimulate the heart. Cardioinhibitory, those that inhibit it. And then the vasomotor center, it's kind of the other aspect of that CV center, is related to the vessel diameter. Okay. And so you can see those things down there are all going to relate to that wonderful autonomic nervous system that we just can't get away from, our sympathetic and parasympathetic divisions. Okay. Now, their activity, or the activity of the cardiovascular center, is not just going to be out of nowhere. It's going to be a direct result of input. Because as we know, feedback, thinking back all the way to the beginning of the semester, we have sensors, or receptors, as we like to call them. Like you can see here, we have proprioceptors, baroreceptors, chemoreceptors, detecting changes, sending a signal into the control center, and then you've got the output, or the motor response. And so we'll talk more about output, but we're also going to talk about the inputs. So we're going to spend the very beginning of Monday talking about baro and chemoreceptor reflexes. We'll also talk about how higher brain centers, and I mentioned this the other day, like your limbic system, your emotional brain, and I used the old scorned ex-lover cheating on you demonstration or analogy, how that can influence your heart rate and definitely also influence your blood pressure. We'll talk a little bit about the cortex and how that can influence blood flow and blood pressure, as can your hypothalamus. I think this is the perfect spot for calling her quits. We've got a couple minutes, so I don't want to get into these reflexes. We won't have enough time to get through them until next week. Everybody have a fantastic weekend.